Hello, welcome again. Today is about double fertilization in plants. The double fertilization in plants, this happening in the flowering plants, whereby the pollen grain, which is from the anthers, will land onto the stigma. The stigma will be producing a sticky substance that will make make that pollen grain, to be sticky. When it sticks on that stigma surface, this pollen grain will germinate to form a pollen tube. This pollen tube will grow down the style and gets nourishment from the style wall. It's getting its nourishment from the style wall. The tip of the pollen tube is a tube nucleus, and the tip is the tubert nucleus. Then this is followed by the generative nucleus. Our generative nucleus will divide mitotically. We form two male nuclei. The pollen tube continues growing down the style into the ovules through the micropyle. When this pollen tube reach our embryo sac, the pollen tube will burst. And this pollen tube now, when it bursts, it gives now way for the tube nucleus again to disintegrate. This gives a way for the two male nuclei to enter the embryo sac onto the ovule. One of the male nuclei, and it's not nucleus, fuse with the egg cell nucleus to form a diploid zygote. So we are saying it's about fusion of one male nuclei with the egg cell nucleus, and we form a diploid zygote, which develops to an embryo. The other male nuclei fuses with the two polar nuclei, and what we get is a deep triploid. It's about three nucleuses now, the two for the polar nuclei and one for the male nucleus. We fuse, we form a triploid nucleus. Not triploid nucleus, but a triploid nuclei. It's supposed to be a triploid nuclei, but not a nucleus, right? Which develops to form an endosperm, but not just an endosperm, but a primary endosperm. And that is what we're calling double fertilization in plants. Thank you, and welcome for other series of our biology sessions. You can click on that subscription button and also ring the bell. Thank you.